When the COVID-19 pandemic began, it was really important to detect all those infected individuals in order to control viral spread. From Irsi-Caixa, we found the necessity of developing a new technology to detect SARS-CoV-2 infection, combining all the good characteristics of both the PCR and the antigen test. And for that purpose, we contact Dr. Valerio Pruneri group at ICFO because they had a lot of expertise in the developing of low-cost biosensors.
The results of our study about the small form factor for fluorometer for SARS-CoV-2 detection is the development of a new and portable device that can detect SARS-CoV-2, that is the virus responsible for the COVID-19 pandemic, from saliva sample in just 30 minutes. Our device is as sensitive as the PCR and as fast as the antigen test.
The rapid antigen tests give a high percentage of false negatives due to their low sensitivity, whereas in our case, we can detect a viral load less than 4,000 copies per milliliter. So we are basically 3 orders of magnitude lower than the rapid antigen tests. In comparison to PCR, since our device is cost-effective and portable, so it can be used anywhere, does not require a specialized lab or a trained personnel to operate.
So our device is built up from very simple components that can be off the shelf, commercially available, so that it's very easy to assemble and is also very easy to scale it up for the mass production.
So we performed a clinical validation of the flow virometry reader, which we did by measuring 54 samples in a blind test, and we were able to reliably detect 31 samples out of 34 positive samples, and with only 3 false negatives. And these gave us a sensitivity of 91% and specificity of 90%. We received saliva samples from Irsi-Caixa Research Institute, and when we received the samples, we first filtered the saliva to remove impurities, and then to this saliva sample we add antibody solution, which is tagged with fluorescence. And if there is a presence of the virus in this saliva sample, the virus will attach to the antibodies, and then we inject this solution into a very small microfluidic channel, and a laser is used to illuminate the sample. So as the solution is flowing, the viral particle, when it hits the laser light, it illuminates a fluorescence and we detect a peak in the signal. So we believe that our flow virometry reader can be used for mass testing of the population, for example, in schools, in sample collection points, in hospitals, and in cinemas or theaters.
As for the future, we would like to test cross-reactivity of such device with other seasonal viruses, for example, influenza or flu, so that we can easily modify it to test other pathogens that are around us. The future possibility is also to develop the kit for self-administration, so that we don't need the personnel to take the sample, or the sample doesn't have to be taken by personnel in hospital, but by user, just spit to the little tube and directly tested by our technology.
Collaborating with Irsi-Caixa Research Institute was very interesting for us because we learned to communicate with them, and in this way we also learned how to communicate with the end user of this technology.